Bombyx mori. Bombyx mori, commonly known as the domestic silk moth, is a moth species belonging to the family Bombycidae. It is the closest relative of Bombyx mandarina, the wild silk moth. Silkworms are the larvae of silk moths. The silkworm is of particular economic value, being a primary producer of silk. The silkworm's preferred food are the leaves of white mulberry, though they may eat other species of mulberry, and even leaves of other plants like the Osage orange. Domestic silk moths are entirely dependent on humans for reproduction, as a result of millennia of selective breeding. Wild silk moths, which are other species of Bombyx, are not as commercially viable in the production of silk. Sericulture, the practice of breeding silkworms for the production of raw silk, has existed for at least 5,000 years in China, whence it spread to India, Korea, Nepal, Japan, and then the West. The conventional process of sericulture kills the silkworm in the pupal stage. 2. The domestic silk moth was domesticated from the wild silk moth Bombyx mandarina, which has a range from northern India to northern China, Korea, Japan, and the far eastern regions of Russia. The domestic silk moth derives from Chinese rather than Japanese or Korean stock. 3. 4. Silk moths were unlikely to have been domestically bred before the Neolithic period. Before then, the tools to manufacture quantities of silk thread had not been developed. The domesticated Bombyx mori and the wild Bombyx mandarina can still breed and sometimes produce hybrids. 5. 342 It is unknown if B. mori can hybridize with other Bombyx species. Compared to most members in the genus Bombyx, domestic silk moths have lost their coloration as well as their ability to fly. 6. Types. Mulberry silkworms can be divided into 3 major categories based on seasonal brood frequency. Univoltine silkworms produce only 1 brood a season, and they are generally found in and around Europe. Univoltine eggs must hibernate through the winter, ultimately cross-fertilizing in spring. Bombyx mori, commonly known as the domestic silk moth, is a moth species belonging to the family Bombycidae. It is the closest relative of Bombyx mandarina, the wild silk moth. Silkworms are the larvae of silk moths. The silkworm is of particular economic value, being a primary producer of silk. The silkworm's preferred food are the leaves of white mulberry, though they may eat other species of mulberry, and even leaves of other plants like the Osage orange. Domestic silk moths are entirely dependent on humans for reproduction, as a result of millennia of selective breeding. Wild silk moths, which are other species of Bombyx, are not as commercially viable in the production of silk. Sericulture, the practice of breeding silkworms for the production of raw silk, has existed for at least 5,000 years in China, whence it spread to India, Korea, Nepal, Japan, and then the West. The conventional process of sericulture kills the silkworm in the pupal stage. 2. The domestic silk moth was domesticated from the wild silk moth Bombyx mandarina, which has a range from northern India to northern China, Korea, Japan, and the far eastern regions of Russia. The domestic silk moth derives from Chinese rather than Japanese or Korean stock. 3. 4. Silk moths were unlikely to have been domestically bred before the Neolithic period. Before then, the tools to manufacture quantities of silk thread had not been developed. The domesticated Bombyx mori and the wild Bombyx mandarina can still breed and sometimes produce hybrids. 5. 342 It is unknown if B. mori can hybridize with other Bombyx species. Compared to most members in the genus Bombyx, domestic silk moths have lost their coloration as well as their ability to fly. 6. Types. Mulberry silkworms can be divided into 3 major categories based on seasonal brood frequency. Univoltine silkworms produce only 1 brood a season, and they are generally found in and around Europe. Univoltine eggs must hibernate through the winter, ultimately cross-fertilizing in spring. Bivoltine varieties are normally found in East Asia, and their accelerated breeding process is made possible by slightly warmer climates. In addition, there are polyvoltine silkworms found only in the tropics. Their eggs typically hatch within 9 to 12 days, meaning there can be up to 8 generations of larvae throughout the year. 7. Larva. Eggs take about 14 days to hatch into larvae, which eat continuously. They have a preference for white mulberry, having an attraction to the mulberry odorant cis jasmone. They are not monophagous, since they can eat other species of morus, as well as some other Morusii, mostly Osage orange. They are covered with tiny black hairs. When the color of their heads turns darker, it indicates they are about to molt. After molting, the larval phase of the silkworms emerge white, naked, and with little horns on their backs. Pupae cocoon. After they have molted 4 times, their bodies become slightly yellow, and the skin becomes tighter. The larvae then prepare to enter the pupal phase of their life cycle, and enclose themselves in a cocoon made up of raw silk produced by the salivary glands. The final molt from larva to pupa takes place within the cocoon, which provides a layer of protection during the vulnerable, almost motionless pupal state. Many other Lepidoptera produce cocoons, but only a few, the Bombycidae, in particular the genus Bombyx, and the Saturniidae, in particular the genus Antheraea, have been exploited for fabric production. The cocoon is made of a thread of raw silk from 300 to about 900 meters, 980 to about 3,000 feet long. The fibers are fine and lustrous, about. Bombyx mori. Paired male above and female below. 5th instar. Conservation status domesticated. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Lepidoptera family, Bombycidae genus, Bombyx species, B. mori binomial name. Description and life cycle: 10 micrometers (0.0004 in diameter. About 2,000 to 3,000 cocoons are required to make 1 pound, 0.45 kilograms. At least 70 million pounds, 32 million kilograms, of raw silk are produced each year, requiring nearly 10 billion cocoons. 8. If the animal survives through the pupal phase of its life cycle, it releases proteolytic enzymes to make a hole in the cocoon so it can emerge as an adult moth. These enzymes are destructive to the silk and can cause the silk fibers to break down from over a mile in length to segments of random length, which reduces the value of the silk threads, although these damaged silk cocoons are still used as stuffing, available in China and elsewhere in the production of duvets, jackets, and other purposes. To prevent this, silkworm cocoons are boiled in water. The heat kills the silkworms, and the water makes the cocoons easier to unravel. Often, the silkworm is eaten. As the process of harvesting the silk from the cocoon kills the pupa, sericulture has been criticized by animal welfare and rights activists. Mahatma Gandhi was critical of silk production based on the ahimsa philosophy, not to hurt any living thing. This led to Gandhi's promotion of cotton spinning machines, an example of which can be seen at the Gandhi Institute. 9. And an extension of this principle has led to the modern production practice known as ahimsa silk, which is wild silk from wild and semi-wild silk moths made from the cocoons of moths that are allowed to emerge before the silk is harvested. Moth. The moth is the adult phase of the silkworm's life cycle. Silk moths have a wingspan of 3 to 5 cm, 1.22.0 in, and a white, hairy body. Females are about 2 to 3 times bulkier than males due to carrying many eggs. All adult Bombycidae moths have reduced mouthparts and do not feed. The wings of the silk moth develop from larval imaginal discs. 10. The moth is not capable of functional flight, in contrast to the wild bee mandarina and other Bombyx species, whose males fly to. Bombyx sinensis more in Hut. Diseases. Instant, she realized. 10 micrometers, 0.0004 in diameter. About 2,000 to 3,000. Bombyx sinensis more in Hutton, 1862. Bombyx texture more in Hutton, 1862. Meet females. Some may emerge with the ability to lift off and stay airborne, but sustained flight cannot be achieved as their bodies are too big and heavy for their small wings. 2 thoracic legs. Adult silk moth. The legs of the silk moth develop from the silkworm's larval thoracic legs. Developmental genes like distalis and extradenticle have been used to mark leg development. In addition, removing specific segments of the thoracic legs at different ages of the larva resulted in the adult silk moth not developing the corresponding adult leg segments. 10. Cocoon of bee mori. Research. A study of an egg of a silkworm from Hooke's Micrographia, 1665. 1679 study of the silkworm metamorphosis by Maria Sibylla Marion. It depicts the fruit and leaves of a mulberry tree and the eggs and larvae of the silkworm moth. Due to its small size and ease of culture, the silkworm has become a model organism in the study of lepidopteran and general arthropod biology. Fundamental findings on genetics, pheromones, hormones, brain structures, and physiology have been made with the silkworm. 11. One example of this was the molecular identification of the first known pheromone, bombicol, which required extracts from 500,000 individuals, due to the small quantities of pheromone produced by any individual silkworm. Many research works have focused on the genetics of silkworms and the possibility of genetic engineering. Many hundreds of strains are maintained, and over 400 Mendelian mutations have been described. 12. Another source suggests 1,000 inbred domesticated strains are kept worldwide. 13. One useful development for the silk industry is silkworms that can feed on food other than mulberry leaves, including an artificial diet. 12. Research on the genome also raises the possibility of genetically engineering silkworms to produce proteins, including pharmacological drugs, in the place of silk proteins. Bombyx mori females are also one of the few organisms with homologous chromosomes held together only by the synaptonemal complex, and not crossovers during meiosis. 14. In the oocytes of bee mori, meiosis is completely achiasmate, lacking crossovers. 15. Even though synaptonemal complexes are formed during the pachytene stage of meiosis in bee mori, crossing over homologous recombination does not occur between the paired chromosomes. 16. Craig Biocraft Laboratories, 17, has used research from the universities of Wyoming and Notre Dame in a collaborative effort to create a silkworm that is genetically altered to produce spider silk. In September 2010, the effort was announced as successful. 18. Researchers at Tufts developed scaffolds made of spongy silk that feel and look similar to human tissue. They are implanted during reconstructive surgery to support or restructure damaged ligaments, tendons, and other tissue. They also created implants made of silk and drug compounds which can be implanted under the skin for steady and gradual time release of medications. 19. Researchers at the MIT Media Lab experimented with silkworms to see what they would weave when left on surfaces with different curvatures. They found that on particularly straight webs of lines, the silkworms would connect neighboring lines with silk, weaving directly onto the given shape. Using this knowledge, they built a silk pavilion with 6,500 silkworms over a number of days. 19. Researchers at the MIT Media Lab experimented with silkworms to see what they would weave when left on surfaces with different curvatures. They found that on particularly straight webs of lines, the silkworms would connect neighboring lines with silk, weaving directly onto the given shape. Using this knowledge, they built a silk pavilion with 6,500 silkworms over a number of days. Silkworms have been used in antibiotic discovery, as they have several advantageous traits compared to other invertebrate models. 20. Antibiotics such as lysosin E. 21, a non-ribosomal peptide synthesized by Lysibacter sp. RH218052, and GPIO363, 23, are among the notable antibiotics discovered using silkworms. In addition, antibiotics with appropriate pharmacokinetic parameters were selected that correlated with therapeutic activity in the silkworm infection model. 24. Silkworms have also been used for the identification of novel virulence factors of pathogenic microorganisms. A first large-scale screening using transposon mutant library of Staphylococcus aureus USA 300 strain was performed, which identified 8 new genes with roles in full virulence of S. aureus. 25. Another study by the same team of researchers revealed, for the first time, the role of YJBH in virulence and oxidative stress tolerance in vivo. 26. Domestication. Gold silkworm, Han Dynasty. The domestic species B. mori, compared to the wild species e.g., B. mandarina, has increased cocoon size, body size, growth rate, and efficiency of its digestion. It has gained tolerance to human presence and handling, and also to living in crowded conditions. The domestic silk moths cannot fly, so the males need human assistance in finding a mate, and it lacks fear of potential predators. The native color pigments have also been lost, so the domestic silk moths are leucistic, since camouflage is not useful when they only live in captivity. These changes have made B. mori entirely dependent upon humans for survival, and it does not exist in the wild. 27. The eggs are kept in incubators to aid in their hatching. Breeding. Silkworms and mulberry leaves placed on trays, Liang Kai's sericulture circa 13th century. Silkworms were first domesticated in China more than 5,000 years ago. 28-29. Pupae. Silkworm cocoons weighed and sorted, Liang Kai's sericulture. Silkworm breeding is aimed at the overall improvement of silkworms from a commercial point of view. The major objectives are improving fecundity, the health of larvae, quantity of cocoon and silk production, and disease resistance. Healthy larvae lead to a healthy cocoon crop. Health is dependent on factors such as better pupation rate, fewer dead larvae in the mountage, 30, shorter larval duration, this lessens the chance of infection, and bluish-tinged 5th instar larvae, which are healthier than the reddish-brown ones. Quantity of cocoon and silk produced are directly related to the pupation rate and larval weight. Healthier larvae have greater pupation rates and cocoon weights. Quality of cocoon and silk depends on a number of factors, including genetics. Hobby raising and school projects. In the U.S., teachers may sometimes introduce the insect life cycle to their students by raising domestic silk moths in the classroom as a science project. Students have a chance to observe complete life cycles of insects from eggs to larvae to pupae to moths. The domestic silk moth has been raised as a hobby in countries such as China, South Africa, Zimbabwe, and Iran. Children often pass on the eggs to the next generation, creating a non-commercial population. The experience provides children with the opportunity to witness the life cycle of silk moths. Genome. The full genome of the domestic silk moth was published in 2008 by the International Silkworm Genome Consortium. 13 draft sequences were published in 2004. 31-32. The genome of the domestic silk moth is mid-range with a genome size around 432 million base pairs. A notable feature is that 43.6% of the genome are repetitive sequences, most of which are transposable elements. At least 3,000 silkworm genes are unique, and have no homologous equivalents in other genomes. The silkworm's ability to produce large amounts of silk correlates with the presence of specific Turna clusters, as well as some clustered sericin genes. Additionally, the silkworm's ability to consume toxic mulberry leaves is linked to specialized sucrase genes, which appear to have been acquired from bacterial genes. 13. In 2018, Illumina's short reads for 137 strain genomes were published. 33. In 2022, Nanopore's long reads for 545 strain genomes were published. 34. Silkworm pupae dishes. Silk moth pupae are edible insects and are eaten in some cultures. In Assam, India, they are boiled for extracting silk, and the boiled pupae are eaten directly with salt or fried with chili pepper or herbs as a snack or dish. Live pupae may be eaten raw, boiled or fried. 35. In Korea, they are boiled and seasoned to make a popular snack food known as byeondaegi. 36. In China, street vendors sell roasted silk moth pupae. In Japan, silkworms are usually served as a sukudani dianzu, i.e., boiled in a sweet sour sauce made with soy sauce and sugar. In Vietnam, this is known as nong tam, usually boiled, seasoned with fish sauce, then stir-fried and eaten as main dish with rice. In Thailand, roasted silkworm is often sold at open markets. They are also sold as packaged snacks. Silkworms have also been proposed for cultivation by astronauts as space food on long-term missions. 37. China. In China, a legend indicates the discovery of the silkworm silk was by an ancient empress named Leizu, the wife of the Yellow Emperor, also known as Shi Lingxi. She was drinking tea under a tree when a silk cocoon fell into her tea. As she picked it out and started to wrap the silk thread around her finger, she slowly felt a warm sensation. When the silk ran out, she saw a small larva. As food. In culture. Instant, she realized this caterpillar larva was the source of the silk. She taught this to the people and it became widespread. Many more legends about the silkworm are told. The Chinese guarded their knowledge of silk, but, according to one story, a Chinese princess given in marriage to a Khotan prince brought to the oasis the secret of silk manufacture, hiding silkworms in her hair as part of her dowry, probably in the first half of the 1st century AD. 38. About AD 550, Christian monks are said to have smuggled silkworms hidden in a hollow stick out of China, selling the secret to the Eastern Romans. Vietnam. According to a Vietnamese folk tale, silkworms were originally a beautiful housemaid running away from her gruesome masters and living in the mountain, where she was protected by the mountain god. One day, a lecherous god from the heaven came down to earth to seduce women. When he saw her, he tried to rape her, but she was able to escape and was hidden by the mountain god. The lecherous god then tried to find and capture her by setting a net trap around the mountain. With the blessing of Guanyin, the girl was able to safely swallow that net into her stomach. Finally, the evil god summons his fellow thunder and rain gods to attack and burn away her clothes, forcing her to hide in a cave. Naked and cold, she spit out the net and used it as a blanket to sleep. The girl died in her sleep, and as she wished to continue to help other people, her soul turned into silkworms. Feeding. Bombyx mori is essentially monophagous, exclusively eating mulberry leaves, Morus spp. By developing techniques for using artificial diets, the amino acids needed for development are known. 39. The various amino acids can be classified into 5 categories. Those which, when removed, cause larval development to stop entirely: lysine, leucine, isoleucine, histidine, arginine, valine, tryptophan, threonine, phenylalanine, methionine. Those which, when removed, impede later stages of larval development: glutamate and aspartate semi-essential amino acids, with negative effects that can be eliminated by supplementing with other amino acids. Proline, ornithine can be substituted. Non-essential amino acids that can be replaced through biosynthesis by the larva: alanine, glycine, serine. Non-essential amino acids that can be removed with no effect at all: tyrosine. Diseases. Beauveria bassiana, a fungus, destroys the entire silkworm body. This fungus usually appears when silkworms are raised under cold conditions with high humidity. This disease is not passed on to the eggs from moths, as the infected silkworms cannot survive to the moth stage. This fungus, however, can spread to other insects. Grassery, also known as nuclear polyhedrosis, milky disease, or hanging disease, is caused by infection with the Bombyx mori nucleopolyhedrovirus, aka Bombyx mori nuclear polyhedrosis virus, genus Alphabaculovirus. If grassery is observed in the chalky stage, then the chalky larva must have been infected while hatching or during chalky rearing. Infected eggs can be disinfected by cleaning their surfaces prior to hatching. Infections can occur as a result of improper hygiene in the chalky rearing house. This disease develops faster in early instar rearing. Pebrine is a disease caused by a parasitic microsporidian, Nosema bombycis. Diseased larvae show slow growth, undersized, pale and flaccid bodies, and poor appetite. Tiny black spots appear on larval integument. Additionally, dead larvae remain rubbery and do not undergo putrefaction after death. N. bombycis kills 100% of silkworms hatched from infected eggs. This disease can be carried over from worms to moths, then to eggs and worms again. This microsporidium comes from the food that the silkworms eat. Female moths pass the disease to the eggs, and 100% of silkworms hatching from the diseased eggs die in their worm stage. To prevent this disease, eggs from infected moths are ruled out by checking the moth's body fluid under a microscope. Flatcherry infected silkworms look weak and are colored dark brown before they die. The disease destroys the larva's gut and is caused by viruses or poisonous food. Several diseases caused by a variety of funguses are collectively named muscardine. Sericulture. Sericulture, or silk farming, is the cultivation of silkworms to produce silk. Although there are several commercial species of silkworms, the caterpillar of the domestic silkmoth is the most widely used and intensively studied silkworm. This species of silkmoth is no longer found in the wild as they have been modified through selective breeding, rendering most flightless and without defense against predators. Silk is believed to have first been produced in China as early as the Neolithic period. Sericulture has become an important cottage industry in countries such as Brazil, China, France, India, Italy, Japan, Korea, Russia, and Thailand. Today, China and India are the two main producers, with more than 60% of the world's annual production. Court ladies preparing newly woven silk by Zhongzan, 713 to 755, reproduced by Emperor Haozong in the 12th century. History. According to Confucian text, the discovery of silk production dates to about 2700 BCE, although archaeological records point to silk cultivation as early as the Yangshou period, 5000–3000 BCE. 1. In 1977, a piece of ceramic created 5400–5500 years ago and designed to look like a silkworm was discovered in Nankyun, Hebei, providing the earliest known evidence of sericulture. 2. Also, by careful analysis of archaeological silk fiber found on Indus civilization sites dating back to 2450–2000 BCE, it is believed that silk was being used over a wide region of South Asia. 3. 4. By about the first half of the 1st century CE, it had reached ancient Khotan. 5. By a series of interactions along the Silk Road. By 140 CE, the practice had been established in India. 6. In the 6th century CE, the smuggling of silkworm eggs into the Byzantine Empire led to its establishment in the Mediterranean, remaining a monopoly in the Byzantine Empire for centuries. Byzantine silk. In 1147, during the Second Crusade, Roger II of Sicily 1095–1154 attacked Corinth and Thebes, two important centers of Byzantine silk production, capturing the weavers and their equipment and establishing his own silkworks in Palermo and Calabria. 7. Eventually spreading the industry to Western Europe. Traditional Chinese process. The silkworms and mulberry leaves are placed on trays. Twig frames for the silkworms are prepared. The cocoons are weighed. The cocoons are boiled and the silk is wound on spools. The silk is woven using a loom. The silkworms are fed with mulberry leaves, and after the fourth molt, they climb a twig placed near them and spin their silken cocoons. The silk is a continuous filament comprising fibroin protein, secreted from two salivary glands in the head of each worm, and a gum called sericin, which cements the filaments. The sericin is removed by placing the cocoons in hot water, which frees the silk filaments and readies them for reeling. The silk is woven using a loom. The silkworms are fed with mulberry leaves, and after the fourth molt, they climb a twig placed near them and spin their silken cocoons. The silk is a continuous filament comprising fibroin protein, secreted from two salivary glands in the head of each worm, and a gum called sericin, which cements the filaments. The sericin is removed by placing the cocoons in hot water, which frees the silk filaments and readies them for reeling. This is known as the degumming process. 8. The immersion in hot water also kills the silk myth pupa. Single filaments are combined to form thread, in a process called throwing, which is drawn under tension through several guides and wound onto reels. This process of throwing produces various yarns depending on the amount and direction of the twisting. 9. The threads may be plied to form yarn, short staple lengths are spun. See silk noil. After drying, the raw silk is packed according to quality. Production. Sustainable silk. Piece silk. The most popular substitute for traditional silk is piece silk, also known as ahimsa silk. The primary factor that makes this form of silk more ethical is that moths are permitted to emerge from their cocoons and fly away prior to boiling. It denotes that no pupa is ever cooked alive during manufacture. However, domesticated silkworms used to make silk have undergone thousands of years of selective breeding and are not manufactured to emerge from their cocoons. They are unable to defend themselves against predators since they cannot fly or see clearly. They typically die soon after emerging from their cocoons as a result. 10. Wild silk. The cocoons of tusser silkworms, which are found in open woodlands, are used to produce wild silk, also known as tusser silk. Compared to conventional silk, their cocoons are typically picked after the moths have emerged, making it a more ethical option. Because wild silkworms consume a variety of plants, their fabric is less uniform but more robust. The fabric is made with fewer chemicals as well. The pupae are still inside the cocoons when they are harvested by certain enterprises that employ wild silk, though. 11. Stages of production. The stages of production are as follows. 1. The female silkmyth lays 300 to 500 eggs. 2. The silkmyth eggs hatch to form larvae or caterpillars, known as silkworms. 3. The larvae feed on mulberry leaves. 4. Having grown and molted several times, the silkworm extrudes a silk fiber and forms a net to hold itself. 5. It swings itself from side to side in a figure 8 feet, distributing the saliva that will form silk. 6. The silk solidifies when it contacts the air. 7. The silkworm spins approximately 1 mile of filament and completely encloses itself in a cocoon in about 2 or 3 days. The amount of usable quality silk in each cocoon is small. As a result, about 2,500 silkworms are required to produce a pound of raw silk. 12. 8. The intact cocoons are boiled, killing the silkworm pupa. 9. The silk is obtained by brushing the undamaged cocoon to find the outside end of the filament. 10. The silk filaments are then wound on a reel. One cocoon contains approximately 1,000 yards, 910 meters, of silk filament. The silk at this stage is known as raw silk. One thread comprises up to 48 individual silk filaments. Mahatma Gandhi was critical of silk production based on the ahimsa philosophy, not to hurt any living thing. He also promoted ahimsa silk, made without boiling the pupa to procure the silk and wild silk made from the cocoons of wild and semi-wild silkmyths. 1314. The Human League also criticized sericulture in their early single, Being Boiled. The organization PETA has also campaigned against silk. 15. Pupae is food. Beyondigi. The conventional method of silk production results in approximately 8 kilograms of wet silkworm pupae and approximately 2 kilograms of dry pupae per kilogram of raw silk. 16. This byproduct has historically been consumed by people in silk-producing areas. 16.